on to session 8, which is integration of digital and AI into biotechnology. And our first speaker is Dr. Andrew Currin from Imperagen. Andrew.
Morning, everybody. I'm Andy Currin, so I'm one of the founders and Chief Scientific Officer at Imperagen. So we are a spin-out. We spun out from the University of Manchester just under 3 years ago now. So yeah, it's great to be here, great to meet some old colleagues and some new colleagues as well. So yeah, really excited to give you a bit of an overview of what we've been up to in the last few years. And so the talk generally will be, I'll start off with give you a bit of an overview of the technology and the platform that we've developed, particularly around some of the digital technologies and the tools that we've developed, and then finish off with some case studies of actually the platform in action and how we've actually engineered some enzymes for kind of real world applications. So first, before we get into the technology, why enzymes? And I don't think I need to say too much about the value of enzymes to this crowd. I won't, I think everybody appreciates just how remarkable they are as catalysts. But I just wanted to highlight that they are adopted already in a large number of different market sectors. So they are a technology that is being used, and we feel they can be used a lot more. But yeah, whether it's in the chemical manufacturing space, very often in the production of fine chemicals and high-value chemicals, enzymes already have a really good standing in their use. So whether it's chemical manufacturing, in consumer goods, everything from biological washing powder up to high-value pharmaceutical APIs, enzymes are being used, and all the way up to some of our kind of bigger kind of grand challenges, which are to do with circular economy and sustainability. Enzymes have a key role to play in that. So they are a known technology commercially, and very often their value is around the ability to control chirality and their ability to either introduce chiral centers or act on specific enantiomers within a mixture. Enzymes have a fantastic ability to do this, and very often their selectivity is one of the key benefits that people look for when they're looking for using an enzyme. So this stereoselectivity, being able to do a specific biotransformation, and very often within a complex mixture, if you think what happens within a cell, there's a lot of other molecules there. It can do a very specific chemical reaction within that mixture. That selectivity is gold dust really for some applications, and it really gives enzymes a big advantage over more conventional synthetic chemistry routes. But they also have, I think Anthony earlier gave a longer list of potential advantages for using enzymes. Another one really is to do with their selectivity. They often offer the opportunity for more efficient, shorter synthesis routes compared to synthetic chemistry. The example I've got in mind is we ran not talking about today, but we ran a case study where actually using an enzyme cut out a series of other extra chemical steps that you would need to use, specifically using protection groups like bot protecting groups. And that really is because they can work with such a level of selectivity that you can actually shortcut a lot of the synthesis routes. So a lot of the benefits in terms of the actual efficiency of the process. And we've talked a little bit about sustainability as well. I think, not unpacking it too much, sustainability can come in in a number of different forms. Very often enzymes offer a sustainable solution to replace, we've heard before, petrochemical routes. But sustainability in terms of replacing highly polluting kind of petrochemical synthesis pathways is important. But it's such a broad topic that I think I don't really want to unpack that too much. So I've given you a flavour actually that enzymes are very valuable molecules, and we heard great talk earlier from Anthony about the fact that you can engineer enzymes, and actually this is a key part of developing an enzyme for a new application, because what you often find is that taking an enzyme from nature and putting it into a new commercial application, particularly in industrial kind of harsh conditions, you might be able to do the chemistry, but it might not be optimal, and you might actually need some better performance improvements from that enzyme to actually make the process even commercially viable. So you can achieve this through engineering, and we had a great talk earlier, so I won't go too much, but I'll pick up on one thing Anthony said right at the end of his talk very quickly. It's like, well, engineering is great, but the process is slow, and it's time consuming, and it's laborious, takes a lot of resources. And this is a real barrier, particularly for commercial adoption of enzymes. That most people want an optimized enzyme, an engineered enzyme, but the cost and the time it takes to achieve that and obtain that is very often prohibitive, and that really is limiting a lot of the uptake of using enzymes. So Imperagen really want to see a wider adoption of enzymes in new sectors, and being able to provide optimized enzymes more quickly and reliably is a real kind of focus of what we've been doing since we spun out. So, as I say, the process is largely slow and unpredictable, and this is one of the barriers. So I didn't want to get onto too much technical information. I've kind of gone for a zoomed out kind of high level view here. This is on the left, I don't know if the pointer's working, on the left here, this is literally a all atom view of an enzyme, just an example enzyme, and this is actually one of the IREDs that we've used in the case studies later on. So I've shown every atom here, and this enzyme is a dimer, and I've highlighted the substrate bound in there, if I can highlight the substrate bound in pink, and this enzyme actually uses a cofactor as well, extra bit of complexity, so the cofactor is actually bound in there. There's 10,000 atoms in this representation, and what we're looking to do, in a nutshell with engineering, is rearrange the order of these atoms to make the enzymes work more effectively. So you can see just visually how complex this problem is. And there are a number of challenges to why it's so complicated, but I've boiled it down to really two key challenges that we can talk about in the talk. There are more, very happy to speak at length about the others, but I've really boiled the problems down to two areas, which is how on earth do you design rationally changes to this enzyme to make it better? So the design process is complex and unpredictable. I could use a number of different adjectives rather than complicated, unproductive, random in some ways. The design process is very difficult. But when you have these designs, what do you then do? You actually go into the lab and test these. The testing process of actually finding improved enzymes amongst all these possible sequences that you could test is a very long and drawn out, largely manual process. There's you can use liquid handlers, colony pickers, but actually the core process is manually driven, and it's quite inefficient. And this is something Imperagen have really focused on. If we're going to address the barriers to using enzymes, we wanted to address these things. So our approach is really to digitalize as much of this process as is possible. So we've done this to really be able to work with better speed and efficiency throughout the whole process. This is not just a series of design tools. We've looked at the whole process and say, how can we work much more effectively, quicker, and with more reliability? So it's really addressed, we're going to highlight these two areas. So it's replacing a largely ad hoc, complicated design process with targeted, specific designs that are more productive than currently there. And we really use AI as not the only technology, but a core technology of doing this design process. And I'll go into how we do that in a moment. But also automating and improving the speed that we can run lab experiments as well. So actually replacing a lot of manual experiments with high throughput end-to-end robotics is something that we've worked really hard on developing. So I just wanted to highlight at this point, you can do engineering for a number of different objectives, and very often when you've started handling enzymes, you probably know kind of what the challenges are and what might need optimizing. But yeah, you can do anything from engineering an enzyme from having a very low yield up to a high yield. You can also address the cost contribution. So that's one of the case studies we'll have in a moment, is the cost of using enzyme is very often prohibitive. You can engineer an enzyme to reduce cost. And you can also, as we heard earlier, engineer enzymes for completely new chemistry, something new to nature that has never been seen before. You can engineer these features into an enzyme. So it's a hugely powerful technology. So that's really, the potential is great, but the access and the speed that you can actually reliably do this at is often prohibitive. But this is something that we've worked hard to address. I'm just going to skip on. It's not skipping on. Great, thank you. So that's largely our problem and our solutions. I just wanted to first give you an overview of a lot of the technology that we've developed at Imperagen. So as you can imagine, the digitalization of a process is going to involve a lot of computational development and technology. So this slide really highlights the computational tools that we've developed. And before I go into some of the details of it, I really want just to highlight that these that these are integrated tools. These are not a series of kind of scripts that are run in isolation. These have been built in a package, and it's all accessed through our kind of custom web app interface. So actually, the access to this is for everybody at Imperagen. It's the computational tools or the lab people all go to this front end web app to run their processes. So it's whether running the algorithms or designing experiments or bringing the data back in, all done within this integrated system. And the integration is really key, and it's something that is hard to do. And I think the closer you get to actually doing this work, and the people that have done enzyme engineering before will see the benefit of actually integrating a lot of this technology together. So that is something we've worked really hard on doing. So starting on the left here, we've developed a number of different algorithms and tools in the design process of, where in the enzyme do we need to mutate to actually find an improvement. And these are tools specifically designed, they are targeting outside of the active site. So actually, it's pretty routine to target the active site when you want to actually look at novel activity. But outside the active site, where on earth do you go and mutate? We have a lot of tools to do that, and I have a following slide, a bit more depth about that. So I won't dwell too much on this slide, but really this is shortlisting from a vast number of possible sequences. Where in the sequence are you going to go into the lab and mutate? Once you have these designs, then you translate that back into a genetic design. So we actually, directed evolution has a lot, really begins at the genetic level. So actually translating those designs back into DNA designs. We design our own genes. We have our own algorithm for optimizing the genetic design of those genes, and also the automatic design of all the DNA oligos. We actually create the mutations that we've designed. So we've created a lot of tools that allow us to very quickly design the experiments. And these go straight into our experimental designs. So the idea here is not that we completely take all the decisions away from the scientists that are going to be doing the work, but we put at their fingertips essentially, all the tools that they need to implement the designs very quickly. So the design steps, whereas in previous life at the university would have taken hours, potentially days in terms of designing these experiments, these have been done in minutes. And actually some of the robotics work you'll see coming up, the experimental design on all the oligos were designed in about 10 minutes. So really the speed and efficiency that we can work using this technology is fantastic. And like I've alluded to, actually programming the robotics is something that we've just brought online within the software. So actually automatically designing experiments for the robotics from the web app is something that we can do, and actually integrating that is a very recent development for us. So I wanted to highlight as well, down the bottom here is data management. Might sound like one of the least exciting of these parts of the system, might be a bit dry, but actually I want to kind of emphasize how crucial data handling and data management is, particularly when you're working at high throughput. And really, if you want to be looking at anything to do with AI, how you handle data, how you label data, the metadata that you save alongside the actual raw data is hugely important. It's something we've put a lot of effort into developing a process for, and we're continuing to refine, but it's not something to be overlooked. That's the management and handling of data is hugely important. So this, like I say, this is all handled within our software. So like I say, the design process is really key to the enzyme evolution process. So I've just got a slide here about our various methods of doing it. So the key one initially is our proprietary way of modeling reaction chemistry. So we actually have a quantum mechanics physics-based algorithm that models the actual transition state for an enzyme. It can do this very rapidly. So actually that's something that is unique, and actually means that we can cycle through in high throughput thousands and thousands of variants and simulate the chemical reaction in the transition state for that variant. It allows us to work at a very high throughput. So for any given enzyme, we'll model that reaction. It's specific for that enzyme, that compound substrate that we're looking at. So it's a very targeted chemical, it's very specific to the chemistry that we're looking at. We simulate millions of mutation combinations, we generate a large in silico data set, and we actually use that to train at the outset an AI model, which is then used to design our first set of experiments. And I've kind of used the phrase zero shot here. It kind of crops up a few times when you're dealing with kind of AI kind of techniques. Zero shot in this context is really from scratch predictions with no prior understanding. So without any actual experimental data to guide you on what works and what doesn't, providing a set of designs is without any prior data is the zero shot element. So when I say zero shot, it's really round one with no prior understanding. How do you generating a set of predictions? So this is an example of one of the models that we've trained purely using the QM in silico data, and we use this to do our first round predictions. We have a number of different algorithms as well. We don't just rely on this. We have a number of different kind of orthogonal approaches. One of them more recently is the integration of language models. There's a lot of interest at the moment in large language models in biology and in day-to-day life. I'm sure most people here have probably used ChatGPT, large language models, but protein language models are a good way of accessing predictions, and they're largely generic predictions. They're not. It's one thing that sets our kind of QM kind of simulation approach apart, is that that's a very targeted chemical prediction method. These are quite generic predictions, but we have it within the platform and we can use it for certain things. Well, one thing we've done is done a benchmarking exercise where we've actually said, how do our predictions compare to the existing kind of large language models, which is generating a lot of interest at the moment. So what we did was a kind of a side-by-side study, the same enzyme, same substrate. It's actually the same round of engineering. We tested both the 30 best predicted variants from the large language model and our best scoring QM variants, kind of compared them alongside each other. And what was really encouraging for us is, whilst the large language model gave some improvement, a lot of the variants, the majority of the variants were around wild types activity. So they were still folded, they were still active, but not a lot improved. A few variants came up to about twofold, if the pointer is working, about twofold improvement, a couple there. But it was a small number out of the 30 that we tested here. And there were a large number of actually inactive mutants too. What we were encouraged by is our more targeted chemical approach, the QM method. Actually, we saw a much higher hit rate, and actually all of our best variants were at least twofold better than where we started. Some moved up to threefold. And that was really encouraging. Not that the predictions were good, but actually for the same amount of work, we can get a lot more improvements. We can build a lot bigger data set. So what we do on a typical round of engineering is we don't just try and look for one single hit and then iterate the process. We try and build a sequence activity data set for us to train our models on. And we do that in every single round. And we typically will try to train a model on about 100 to 200 different variants with actually improved activities. These are not kind of largely inactive mutants. If we can train the model on 100 to 200 improved variants, we can train a really good model from that. And we actually use data then to iterate the process, and there's some examples in the case study later on in terms of how we use trained models and data to train the model to move more quickly. So then I want to move on to having had a set of designs, actually moving into the lab and testing these predictions. That is a key part of our technology. So actually we've developed a robotic system that runs the end-to-end process from start to finish. So this is our robot in the lab. It's running one of the experiments. And what we found is we developed a lot of the methods to work on this. So this is not, the methods that we use are actually very different to a conventional enzyme engineering approach using libraries. We have our own proprietary methods to run this quickly. And we can run experiments in about a week, and that's in a side-to-side. We all right for time? How are we?
Yeah, about 1 minute.
Right, great. So I want to get onto the case studies as well. So I will skip over this, but essentially in a head-to-head comparison, we ran the same experiments between the robotics and manually. We found the robotics was able to work 3 times faster, but actually critically with 5 times less contact time. So whilst a manual process was taking someone full-time over 3 months, in 38 days the robotics was working. Actually for each round it took just 6 hours of hands-on time to run. I'll skip over this, but essentially the benefits of the platform, very happy to speak to anyone outside of this talk, happy to talk through more of that. But I really wanted to give a bit of a case study example of using enzymes, using the platform. So the first one here is a collaboration that we ran with Enzymicals and Xanoprima. So Xanoprima actually created a synthetic route to enantiomer pure nicotine. So I think we all know what nicotine is used for. But they were able to have a process that was actually a much more sustainable and greener synthesis route compared to extraction from tobacco leaves. And so actually, what I was surprised about actually really was how more sustainable this process was. The key element of this is the introduction of this chiral center using this imine reductase enzyme. So the reduction of myosamine into S-nornicotine needed to be very enantioselective, so they're very pure. This was done, commercialized up to multi-ton scale, so the enzyme and product are being produced at ton scale. And so it's a very effective enzyme already. But we were faced with this project, and actually what we were presented with was, whilst it was a very effective process, it had challenges. And it was actually a challenge that a lot of bioprocesses have, which is the cost. The cost of using this enzyme was almost prohibitively too high. The cost of producing the enzyme, adding the cofactors into the bioprocess, the GDH to recycle the cofactor, and that was really a big problem. So we were given this enzyme to engineer, and we were able to, over 2 rounds, first round, 0-shot predictions, we got about a 2-fold improvement, but then training the model based on that experimental data gave us a 10x improvement, and we were really pleased with this, because actually this was a very effective enzyme already. And so these numbers are scale-up values. These are not just small scale from our lab, these are from the enzymicals. These are in scale-up conditions. A 10x improvement was a real result, and actually it seems to be a bit of a game changer in terms of whether this process gets fully adopted up to into further and wider use. Actually, they can drive down the cost of this product because the enzyme activity is that much better. So on a very brief, our final slide actually, so that's the engineering of a very effective enzyme that is engineering to reduce the cost of that, the cost impact of that enzyme. This example is using IREDs for pharmaceutical and high-value pharmaceuticals, so actually using this IRED reaction to produce a chiral API for pharmaceuticals. We were able to, so actually this enzyme did the chemistry, but actually was quite inefficient. We only got a 2% yield from quite a lot of enzyme. What we were able to do was, through a series of rounds of evolution and iteratively training the model on quite a decent amount of experimental data, was to get a significant boost in activity. And actually what's key here is on the structure. We ran a conventional active site kind of engineering process alongside our usual process. The best active site variant we found had about a 9-fold improvement that we obtained in round 1. The variants that we got outside of the active site and were obtained through the QM simulations and training the model to run through rounds 2 and 3 were the most impactful in terms of the enzyme performance. So actually by round 3 we were able to get an over 1,000-fold improvement in the enzyme performance, and we were achieved that by not only just increasing the yield in the reaction, but dropping the enzyme loading as well. So we've gone from 50% loading in this example down to 2%, but we get good yields down to 1% loading now. So using 50 times less enzyme, but maximizing the amount of product yield in the reaction as well. So that was a real good kind of validation of not just having good 0-shot predictions from our algorithms, but being able to train models using experimental data and enough experimental data to get meaningful predictions for a further round. To move through 3 rounds and very quickly get a high yield from a very poor starting enzyme was a really good validation that we could work not just productively but quickly to deliver kind of improved enzymes for API synthesis. So that's about finished up, I think. Yeah. If we're all right for time. Cheers.
Thanks very much. I'm not going to take any questions, but we do have a stand outside. So, yeah, brilliant. Thanks very much. So the next speaker in this session is Cathal, who is also going to be extending what Bob was talking about, utilising machine learning and AI tools to understand difficult to engineer enzymes.
Okay, hi everyone. As I said, my name is Cathal. I'm in the Scotland group at the MIB. I'm a third-year PhD student. The scope of my project's a bit smaller than the previous talkers, but they did cover some stuff, so maybe that I'll cover, so I won't have to do so much of an intro. So the particular enzymes I work on are monoterpene synthases. Monoterpenes are all have the usual kind of characteristics of interesting engineering targets. They produce valuable chemicals. Enzymatic synthesis could offer a green solution to the production, all the same stuff you usually know. Particularly interesting is that you have GPP here, and it can be used to make a range of products. Specifically linear versus cyclical products, and you can see with the development of this, the alpha terpineol cation, you can produce a much more diverse range of products. And that's what my work focuses on. So the problem is, engineering monoterpenes is a pain. Loads of people have done it before me, and there's loads of reasons why it's difficult to do. One of the main ones is low sequence functions relationship. If this was really well understood, then you wouldn't need to do, it would be really simple and you can engineer them quite easily. This also means that they're extremely plastic, where you can make lots of different residue changes, nothing happens to the product profile, and then you can make one residue change and the product profile massively changes, right? So structural based approaches actually have been shown to be really useful for engineering these enzymes. However, they're really difficult to crystallize. So they don't really want to be worked with, to be honest, but we're at a unique point where we can take this as maybe an engineering challenge and see if the tools that we have at the moment are accurate enough to try and finally crack these enzymes, you know? So there's previous work that kind of supports this, where they use for sesquiterpene synthases, which are 15 carbons versus 10 carbons of monoterpenes, and they supplemented their sequence-based model with a homology model data, which helped improve the accuracy and increase the learning from the sequence that they could do. There's also quite a number of monoterpene syntheses already published with their product profile published, so you can take from that. And AlphaFold seems to be quite happy, where Blue's reasonably happy with the accuracy that it predicts these enzymes. Now, when you do a structural alignment of them, there's no, what obviously, as I previously said, I'm looking at the difference between the linear producing groups and the cyclical producing groups to try and work out how do you get this generation of the alpha terpineol cation and unlock this diversity, right? So unfortunately, I did an alignment and nothing just obviously popped out. And also what you can also see is there's all these kind of extra bits hanging off, which kind of would add noise to the enzyme, and it's shown that within the active site is the important parts and tends to be the hot zones for the things that influence the product profile. Really interesting as well when you do when you look at the active site, it's really highly conserved, and it's only little small changes in the active site that cause the differences. Same thing happens with sequence-based alignments between the cyclical group in green and the cyclical group in blue and the linear group in green. There's no things that immediately pop out, and the same thing for structure-based alignment, likely due to the extra kind of domains that hang off that have no kind of group specificity. So, thought is, can we use machine learning to help understand and hopefully engineer these enzymes? Came up with a workflow. First, used the generated, the previously published data to generate structural database. As we said about the active site is a hotbed for these mutations that cause a change in product profile. So we're going to focus on that to reduce the noise of it as well. Use this as an input for statistical learning and also develop a machine learning model, and then hopefully develop a model to test experimentally. 3 problems with the approach: binary classification model. Enzymes aren't a binary, but that's the only model we can use with this data set size. Computational predictions as well with AlphaFold. I feel like if there's any structural purists here, they might hate me for using AlphaFold, but I think it's kind of time to change on that. And also, this is a big data set for a lot of finding it in literature, and other people have done it, but this is not massive for machine learning standards by any shape or means. Created a data set focusing on these 2 enzymes that produce these 2 as an analog for linear and cyclical, because we want to find out about this alpha terpineol cation generation. carried out statistical analysis on it. From the initial analysis, we can see we can generalize a lot of published data which focused on individual enzymes, where they found that the active site volume, obviously the active site volume of the cyclical products seems to be more highly controlled than the linear, and this is because the linear products just need to ionize the GPP, and then they can be early quenched. And also steric effects have been shown to play an important role, so you can see the amino acid base volume score has an important difference here. And then we can see important differences within the active site that might be useful for engineering later. Also, in terms of aromatic residues, these are known to play an important role in terpene synthesis, specifically different product profiles. Different product profiles. The important takeaway from this is that the type of aromatic residues differ between them, and they're not interchangeable. And this was previously known, and now it's generalized on a wide data set. And in particular, interesting here is the highly conserved single tryptophan here in the cyclical group, which when I looked at in a sequence-based, I wanted to see what typically was in the spot for the ones that didn't have it. However, when I aligned the sequences, you can see here, it's actually around the corner from this in the alpha helices. And it's not a problem with the alignment here. This is the metal binding motif. It's clearly just, there's a there's just a slightly, basically the sequence-based analysis isn't super useful, and this for finding if the residue is conserved in structural space and not in sequence space, right? And this is really bad because structural residue conservation is really important for terpene synthesis. You can't just move a residue and it do the exact same thing. The cations travel down the active site, and this can alter the product profile. So what I did was I made an algorithm which apparently no one else had done before, which is, I thought there would be some off-the-shelf thing, but I guess there's not been enough structures now. I used this algorithm to find whether the residues were conserved in structural space rather than sequence space, because that's what the important part is. Some key takeaways is that this is the one that you kind of needed to look for in it, to make sure everything is okay. And you can see here the DD is conserved between both of them. If that wasn't there, it would have shown us that likely the algorithm doesn't work properly. You can also see things like the highly conserved tryptophan here, and other domains that are likely necessary for catalysis. Like if you knock out this E here, or the serine, you're probably going to knock out terpene catalysis. Also, interestingly, you can see differences between them. So you can see here the tyrosine, and then tyrosine between groups is different. So likely, if you add this, maybe that'll be an interesting engineering target. And here as well, there was no residues within range for most of these, whereas within the cyclical group, there's a histidine there, and when aligned with this a structure that people have done alanine scanning on and mutational analysis on, this histidine was important for cyclization. So that's pretty helpful. We've identified some potential mutation targets that can cause a change in product profile. Also identified some new motifs that weren't previously understood. So the plan is make a machine learning model. However, active site, as Doug said as well, the data set had 50 features, right? 50 factorial different ways of building the model, right? How do you break that down? Obviously, you can't do it, test them all, and most of the features are redundant, and they have hypercorrelation, which you don't want to have in a machine learning model. So what I did was I used a thing called Bayesian optimization to test for the feature space, where it tries to optimize the accuracy output by combining different features within it. I wanted a preference for exploration around this, where this could be like, I don't know, a set of 10 features, this could be a set of like 14 features, different features, blah, blah, blah. And it learns throughout that to find which one's going to lead to the increased accuracy. And through that, we learned a bit about what ones are important for discriminating between linear and cyclical groups. Through that, after like 175 iterations, we managed to find an accuracy of 92%. When we looked at the features within it, there's no hypercorrelation, it's looking all good. And also we did some cross-validation as well, and everything looked fine. So we're happy enough with that. Now that we have the model that's able to discriminate between the groups, the plan is to use it to do ML-guided engineering. How do we do that, right? How do we do this in silico? How do we use this predictive model and turn it into a method for predicting the change in product profile? First of all, find a suitable engineering type, and and use the ML algorithm to find residues that can swap classification. I guess as well, a lot of you are probably asking why not use all these kind of exciting de novo tools. I think, in terms of use in enzymes, typically terpene synthases are purely characterized on PDV, so diffusion and inverse folding models are unlikely to work. And you don't really end up learning loads about the enzyme. Also, they rely on huge data sets, and like philosophically, is the answer to solving any problem throwing loads of data at it, or is there ways around that where you can use the data that's already available, generated by other scientists, to spend less money and obviously less resources in general. So how do we reduce this testing space? Amino acids 20 to the within the active site, even though it's an active site of 29, 20 to the power of 29 is still an insanely large number. Yeah, and so what we want to do is we want to reduce it so that we're able to generate a reasonable amount of variance that can be tested within the model. How I did that was basically not including amino acids that's going to massively change the structure. Took 3 of those out, and then I started using the previous work that I did with the conserved residues, and chose ones that didn't seem like they would just knock out terpene synthase activity. From that, we took down the sequence space to 10 to the power of 12. That's a lot more handy than 10 to the power of 37, and generated 10,000 in silico variants. And from that, we found 240 positives, which were selected for 50 mutants for testing. However, that is where I'm at. So I'm sorry for the cliffhanger. I wish I had the results now. Maybe come back to me in a month or two. But I guess the conclusion of my talk is that terpenes are valuable and just industrially important chemicals for current synthesis. However, they're difficult to engineer, and this offers a good challenge, because if we're able to engineer these quite simply with the data that's available, then this is promising for ones which are less complex. What I've done is built a machine learning model that can predict between product profile between linear and cyclical analogs, and I've developed a workflow for screening in silico mutants for a change in product profile. And some of them, I guess the takeaway as well is data generation is massively changing. This could be with microfluidics, this could be with the fact that now people can generate structures, highly accurate predictions of structures, and ML and data-driven approaches can massively help you in your kind of massively help you in your work to try and understand whatever you're working with. And it's never been easier to learn coding or how to use these tools, but it is difficult to find out which tools to learn. Some of the work that I did with Nucleate has been trying to teach people about how to use AI in protein design. We have an intro talk with someone from the Baker Lab about it. It was an hour long, basically going through their stuff. And then also there's a panel with people, founders from Baker Lab, MIT, somewhere in Canada. So you guys can check it out if you're interested in that as well. It also has a resource page which has loads of resources of which can help you learn about how to use AI for protein design. That's it.
Thank you very much. I think it's always interesting to leave people on a cliffhanger. Any quick questions? One over there.
Thank you. It's probably a question for all the speakers, actually. So you mentioned using Bayesian optimization in your search there, and one of our clients is taking a robot scientist approach to a different problem, cell culture media development. But what they've found is something Doug alluded to at the ProSAR. Your initial sampling really determines the rate at which your search will converge. So you could have like Latin hypercubes or fractional factorials or Sobol sequences. There's loads of ways of starting it. And if you get the start wrong, it'll probably take you longer to get to your answer. So I guess for the whole set of speakers so far, does anyone actually look into this for protein engineering? Or are we just, taking the ProSAR and maybe, some of the techniques that you use, but grounding it in the theory seems to matter a lot in terms of like getting really efficient search.
I actually have a really strong opinion on this. I was at a kind of panel that was talking about pure computational people make better models than a mix of comp and bio, which I just think is ridiculous. It's based off their pure benchmarking. A lot of the stuff relies on you actually knowing about the enzymes, and that's why I think it's important to kind of get yourself in tune with these models, and because they're going to become an increasing part of biology. Because the same way that you need to know the biology, you also need to know the computational approach as well that you're going to be using and you're likely to be using as well. I don't know if that answers your question though.
It does, because the lesson from this client of ours is essentially you need very interdisciplinary understanding.
Yes.
So you need to embed the biology and the computer science theory.
Yeah, 100%.
Brilliant. I think we're going to have to move on, but thank you very much. So our last speaker in this session is Dr. Maribel Montufar from Simplex Molecular.
Good afternoon, everybody. I hope you are hungry, because I am. Looking forward to the lunchtime. So I'm really happy to be here today presenting what we're doing at Simplex Molecular. Simplex Molecular is a startup company based in Manchester. And our mission is to make molecular diagnostics accessible to all. So this talk is going to be about a collaboration that we are currently having to accelerate enzyme discovery for accessible molecular diagnostics. And this is, again, in collaboration with another startup called FastFall, which mission is to make accessible all the computational tools for protein modeling and protein discovery. So I'm gonna start talking about the actual problem that we are trying to target at Simplex Molecular, which is the accessibility to molecular diagnostics. So since COVID, there have been many innovations in regards to molecular diagnostics, new systems, more portable systems to provide the detection of pathogens quicker. However, the reality is that most of molecular diagnostics remains centralized. And there are many communities around the world that don't have access to these high-tech laboratories or high-tech expertise. And some of these communities, can you see that? Yeah. So they still rely on small machines and actually like solar panels to make sure that they have the power supply required to run this equipment. So, so another problem that the centralized molecular diagnosis is the fact that reagents require cold chain transportation. They also need to be kept in cold environments. So some areas in which they don't have the access to, electricity in fridges and cold storage, they have this problem. Oh, there you go. It's working now. Another problem is that some of these platforms that are used for current molecular diagnostics, like PCR, LAMP, they require thermocyclers or some other instruments to maintain temperatures, so the actual methodologies work to detect whatever they want to target. And also they need to have readout systems to have this detection of fluorescence, luminescence, or any other detection they might need to actually validate if the test is positive or negative. So at Simplex Moleculars, we thought, like, what if we don't have these barriers in infrastructure? The approach that we're having is that we want to design and manufacture robust reagents based on recombinant enzymes to enable cold chain freedom. So it means that the reagents can be transported without a need of any cold chain, and also that they can operate at ambient temperatures or at room temperature without the need of machines or power supplies. This is our actual vision of how we envision our product, which is reagents integrated in microfluidic devices in which we can have multiple reactions working at the same time, and really similar to what a lateral flow test works, so in which we can just add the sample, mix, and wait for a color development to know if the reaction is positive or negative. So this could be really useful for remote areas in which they don't have access to laboratories and they need to detect quickly for pathogens or genetic diseases or any problems that might arise in the future. So we know that to achieve this is a really complex path, and that's why, oh my God, that's a mess. Wow. Okay, well, that's why we are leveraging our collaborations. So as we are based in Manchester, one of the main collaborations is through the University of Manchester, as they provide access to experts and world-leading facilities for protein optimization. But more recently, we have started a collaboration with this company called Fastful, which again, they aim to democratize the access to computational tools for scientists that are not necessarily bioinformaticians, and they don't know exactly in depth how to use those tools. So hopefully with this collaboration, we can make this development of molecular enzymes a bit faster. So we started by designing this case study in which we are designing a thermostable helicase. So helicases are really useful to unwind the DNA strands. So in case of molecular diagnostics, we can kind of separate the strands and try to target the region that we need to. In case of a pathogen, for instance, COVID, I will put as an example. But usually for current methods, the way that we unwind these double strands is by using temperature. So we want to use now enzymes. The pipeline we are creating is by, oh my God, this one. First, to characterize helicases with a candidate selection. Then the FastFold is incorporating the AlphaFold tool that we already know. So this is to predict 3D structures of candidate proteins. Then we will integrate another tool that is called EvoDiff to generate a variety of functional sequence variants. Then we're gonna integrate this thermodynamic prediction system to understand how stable will the enzymes be. And finally, the GrowMax, which will help us with the validation of the structural performance of this enzyme. So I just want to clarify, the FastFold platform basically is like a ChatGPT platform that integrates different ML systems that are openly available, but usually that requires a lot of knowledge of bioinformatics so the scientists can be users. So the idea of FastFold is to integrate all these open source coding tools in a ChatGPT platform in which we can ask and interact with the agent and ask questions about, okay, is this protein functional? What can I change with this protein? Can you tell me more about the characteristics, the functionality, et cetera, very quickly in minutes. So at the moment, we are still in the process of developing the whole pipeline, but this is just a screenshot of how the actual platforms looks like. So we can give a sequence and ask the platform to just fold it for us, and immediately it gives us a structure, and with all the predictions, just like AlphaFold does. This is a completely 3D. We can just move and select any amino acids we want. We can also ask questions about the protein in question, like what is the function, and it's going to give you, based on the actual structure, what is, and based on some data search that the platform is connected to different databases, what is the function of the protein. You can ask about highly conserved regions, and it's going to tell you what are the regions, what are the, if you want to create mutations, what are the regions that you should not touch or that you should not mutate in order to don't affect the activity of the protein. Many questions, again, about how flexible the protein is in comparison with a wild-type protein. So you can interact with this agent, as I said, like a ChatGPT. Give you plots about the interaction when you have these enzymes interacting with other molecules, like DNA in this case. Again, this works in seconds. You don't need to know anything about bioinformatics. Of course, at the end, you will need to interpret what this data means, but at least as a first shot to understand, is this worth exploring a bit more? It will be really good tool for everybody that wants to explore a little bit on initial proteins. And again, the platform is connected to different databases. So if you submit a protein that is already been reported by anybody, it will tell you, because it's going to do a deep search in the web. So it will tell you everything that has been done to the protein. So you already know, like, okay, this is already been done. I'm not going to try this. And it's very quickly, again, in minutes, you can have these results. So yeah, basically that's what we've been doing. Again, we are continuing building the pipeline. So we hopefully can just ask the platform, can you tell me which amino acid to mutate in order to make it more stable, to make it more highly processive, to change the processivity, and try to address these problems with computational models more quickly. And again, the idea is to democratize these tools so anybody can have the use of this as soon as possible. Just to conclude the talk, both companies, we have this idea of democratizing. Simplex Molecular is interested in democratizing the access of molecular diagnostics. Fastfall is with the mission of democratizing the access to molecular tools for computational biology. And if you want to have a look of what we're doing, I invite you to scan the codes. You can access the Fastfall platform. It's free to use, and you can start interacting and submit your proteins and generate your models if you want. And they are open to collaborations. They started with validations of their models in drug discovery, and now they are trying to validate their platform with different uses, in this case, protein optimization, which is a bit more complicated. So yeah, feel free to reach out just to me or through the QR codes. And thank you. Any questions?
Any questions for Maribel? Doug?
So if you run AlphaFold 100 times, you get 100 slightly different answers. And actually the noise in those answers can be used to give you a poor man's molecular dynamics. Yeah. If you run FastFold 100 times, do you always get the same, or do you get differences which can also give you useful information?
You mean on the platform here?
Yeah.
I've seen that there's no difference once I run, again, the same sequence every single time. And if I ever see a difference, it's literally by a centimal, like in the prediction. It's not really high variability.
Then in that case, we'll call time because we're already a bit overrunning the session. And thank you to everybody for talking.
Thank you.